Hello everyone. So today we are going to discuss our third unit. As we have completed our last two units in the previous classes, now we are going to start our third unit, that is regarding the immunity and immune reactions. So before going to this topic, we must know what are the important immune cells which are mainly involved. Okay. So it mainly involves antigen and antibody reactions. So before going to know what is that reaction and what are the cells involved in that, so first we must know what is antigen and what is antibody. So antigen and antibodies, both of these are like protein molecules. So antigen is a foreign particle which invades our body, and in order to counteract that, antibody will be produced. So antigen is defined as a substance which is usually proteinic in nature. So which when introduced into our body, antibody production will be stimulated. And coming to antibody, antibody is also a protein substance which is produced as a result of antigenic stimulation. So only when there is presence of antigen, this antibody will be produced. So mainly the circulating antibodies are like in the circulation, what are the antibodies that are present? They are like immunoglobulins like IgG, IgA, IgM, IgE and IgD. So what are the circulating antibodies are there? That we call them as immunoglobulins. Okay. And what is hapten? Hapten is nothing but like in a chemical reactions, how does vector acts? The same thing will be with this hapten. Hapten is a non-protein molecule. It does not have any antigenic property, but what does it do is it will when it combines with an antigen, it forms a complex which is capable of forming antibodies. So when it binds with other antigen, it will have a antigenic property. Basically, it is not an antigen, but when it binds with another antigen, it will have these antigenic properties. Okay. So what is the primary reaction which takes place? So whatever the antigen is there, when it comes and binds with the antibody, it forms a complex that we call it as antigen antibody complex. So this we consider it as a primary reaction, and then the secondary reaction is whatever this complex is formed, whatever the processes it will undergo in order to cause this tissue damage. So that we call it as secondary reaction. It includes agglutination, precipitation, immobilization, neutralization, lysis, complement fixation. So these reactions will, this antigen antibody complex will undergo these kind of reactions, and finally it causes tissue damage. So this is all about antigen antibody and how does it cause antigen antibody complex formation. Okay. And then coming to types of immunity. So first, what is immunity? Immunity is nothing but what is the defense mechanism which is present in our body in order to fight against these foreign particles. That we call it as immunity. So in immunity, again, it is of two different types. One is natural immunity or innate immunity, and the other is specific or acquired immunity. Okay. So what is natural immunity? Like it is non-specific. Already our body will be armored with itself with some kind of properties, with some kind of antibodies in order to fight against the upcoming antigens. So it is considered as a first line of defense, and there is no proper antigenic specificity, and it is non-specific. So it has two components. One is humoral component and the other is cellular. In humoral, it consists of complement molecules, and in coming to cellular components, it includes neutrophils, macrophages, and natural killer cells. And coming to specific or adaptive immunity, so the name itself indicates that it is specific. So that means this immunity, that means these antibodies will be produced in response to specific antigens. So it will have antigenic specificity. So that is the reason it is called as specific or adaptive immunity. Okay, it also has two main components. First is humoral, the other is cellular. In humoral component, the cells involved are antibodies that are produced by B cells. In cellular, it is mediated by T cells. So B cells, T cells together comprise of lymphocytes. So these are two different types of lymphocytes. So before going to that, we must know what are the different cells are involved, which are involved in the immunity. Okay. So what are different cells that are involved in immunity? Lymphocytes. So in lymphocytes, again, T lymphocytes and B lymphocytes, monocytes and macrophages, mast cells, basophils, neutrophils, and eosinophils. So these are the different types of cells which are involved in the immune reactions. So we'll be seeing in detail what are the different roles of each kind of cells. Okay. Lymphocytes, it includes like 20 to 50% of immune reactions will be done with the help of these lymphocytes. Again, it is of two different types, B cells and T cells. So B cells comprise 10 to 15% and remaining 75 to 80% is comprised of T cells. Again, B cells are again of, they will be transformed into plasma cells and will participate in the formation of immunoglobulins, which will directly participate in the immune reactions. And coming to T cells, again, these are of three different types, T helper cells and T suppressor cells, and the other is natural killer cells. So all these will promote cell-mediated immune reactions. And coming to monocytes and macrophages, so these helps in the recognition of this antigen and they help in the phagocytosis and antigen presentation. So these are the functions of monocytes and macrophages. And coming to mast cells and basophils, these are mainly involved in allergic reactions and also in wound healing. And eosinophils are also involved in allergic reactions. That means these levels will be elevated in the allergic reactions. And neutrophils are mainly seen in, these are the first line of immune cells, which will fight against the microorganisms, whether it may be like bacteria, viruses, whatever it may be. They are the first line to fight against these microorganisms. Okay. So this is all about different types of cells of immune system. Mainly lymphocytes, monocyte macrophages, mast cells and basophils, neutrophils and eosinophils. So these are the cells of immune system. And then coming to, before going to this table, we'll be seeing this, we'll be going to a small note on B cells and T cells. As I said, the T lymphocytes are lymphocytes are of two different types classified into B cells and T cells. So B cells will be composed, they'll be comprised of 10 to 15% of lymphocytes. So what does it do? It is involved, B cells are involved in humoral mediated immune reactions, whereas T cells are involved in cell mediated immunity reactions. So how does this B cells will act? So when an antigen contact with these B cells, what does it, this is an antigen. When it comes in contact with B cell, it's on its surface, it has some receptors, that is B cell receptor it has, and it also has an FC component. So whatever the sticks are there, so this is the FC component which helps in binding of this antigen to the B cell surface. So with the help of this FC component, this antigen comes and binds with this B cell, and once this complex is formed, these will be transformed into plasmacytoid cells. So these FC components will be lost and it will be transformed into plasmacytoid cell, and again it will be converted into plasma cell, and once it is converted into plasma cell, it will cause the production of, it will secrete immunoglobulins. So these immunoglobulins are involved in humoral mediated immune reaction. So this is how B cells are involved. Okay. So it has surface markers like CD molecules. It includes 19, 20, 21 and 23. So these are considered as B cell markers. It has B cell receptors for surface immunoglobulins like IgM and IgG for binding to this antibody molecule. And for B cells, even some kind of T cells like T helper cells will helps in the will helps these B cells in order in participation of these immune reactions. And then coming to T cells, these are mainly involved in cell-mediated immunity and also in delayed hypersensitivity reactions. Majorly, these are divided into two different types, that is T helper cells and T suppressor cells. So, and the other is natural killer cells, mainly T helper cell and T suppressor cells. So these helper cells are also they will be denoted as TH cells, and T suppressors will be denoted as TS cells. Okay. So what does this T helper cells do? They will help in the promotion of this immune reaction. So they are also called as T regulatory cells. They will be having a surface molecule called surface receptor called marker called CD4 molecule. So that is the reason they are called as CD4 plus cells. And when coming to T suppressor cells, they will be having on their surface, they will be having CD8 molecule on their surface. So they are also called as CD8 plus cells. Okay. So what does this do? These T helper cells will not directly cause a cell damage. So first they will elaborate certain cytokines. So these cytokines will release certain interleukins, and these interleukins will be responsible for the immune reactions. So What are the different cytokines that are produced by T helper cells? Again, these T helper cells are of two different types, that is Th1 cells and Th2 cells. So Th1 cells will help in the elaboration of cytokines like interleukin 2 and interferon gamma. IFN is nothing but interferon gamma, and Th2 cells, they will elaborate interleukin 4, 5, 6 and 10. So by formation of these interleukins will participate in the immune reactions. So here T cells are directly not involved in the reaction. So T helper cells will elaborate certain cytokines. Whatever the interleukins that are produced by these cytokines, that will involve in the immune reactions. Okay. So as CD4 plus cells are mainly seen in case of viral infections when these viral antigens are introduced. So at that time CD4 plus cells will be elevated. So that is the reason we'll be checking these CD4 plus levels in case of HIV, and these are also seen in case of tumor destruction. So and like tumor lysis. So in that conditions also we can see the elevation in these CD4 plus cells. And then coming to T suppressor cells, as I already said that they will be having a surface marker called CD8. So that is the reason they are called as CD8 plus cells. Okay. So when coming to CD8 cells or T suppressor cells, they will suppress the immune reaction. So name itself indicates that they will suppress the immune reaction. Then how does it involve in immune reactions? So even though they will suppress the immune reactions, what will they do? They will directly cause a damage of the cell. That is, they will be directly causing damage of the cell which is having this antigen. So whatever the antigen which is having the antigen, like whatever the cell which is having antigen, that cell will be directly damaged by these T suppressor cells. So this is how it will help in the immune reaction. Okay, this will suppress the immune reaction, but it will cause direct cytotoxicity to the cell which is having antigen. So that is the reason they are also called as cytotoxic T lymphocytes. Okay. So these are involved mainly in the cells which are infected with viruses, foreign cells, and tumor cells. And then coming to natural killer cells, like morphologically, they will be completely different from these T helper cells and T suppressor cells. They will be like large. They will be having more amount of granular lymphocytes can be seen. And these natural killer cells are mainly involved in natural immunity or innate immunity. What does they do? The main role of them is to recognize the antigen antibody complex. So whatever the new particle enters, when it forms a complex with this antibody, natural killer cells will recognize the antigen antibody complex and will target and will kill that target directly. So this is how natural killer cells will act. So they will, whatever the targeted thing is, when they will kill directly with the help of a mechanism called antibody dependent cell mediated toxicity. Okay, so its main role is recognition, and once it recognizes, it will kill that cell with the help of antibodies. So that is the reason it is called as antibody dependent cell mediated cytotoxicity. So this is mainly seen in case of, this mechanism is mainly seen in case of the cells which are infected with viruses, and also in tumor cells. This can be seen. The role of natural killer cells can be seen. NK cells can be seen. So here if you see this, the CD markers for B cells is 19, 20, 21, 23. For T cells it is 3, 7 and 2. For natural killer cells it is like 2, 16 and 56. So these are the NK cell markers. So if you see this in picture, this is the Fc component which helps in binding. So in these T cells, this Fc component is not there, that is absent, but it has receptors like T cell with TCR which helps in binding, and it is of two different types, Th1, Th2, Th and T suppressor. Again, this Th is of two types, Th1 and Th2. So Th1 cells will elaborate cytokines like interleukin 2 and interferon gamma. Th2 cells will elaborate cytokines like interleukin 4, 5, 6 and 10. So this red dot is there, no? This is considered as an antigenic molecule, okay, antigenic protein. And these are the T suppressor cells. They will suppress the immune reactions, but they will cause direct cytotoxicity. This is natural killer cell. It does not have any receptors, as we have seen in case of B cells and T cells like TCR, BCR. This is not present in natural killer cells, and their size is large and granulomatous. And what does it do? It will recognize this antigen antibody complex, and it will directly cause the damage of that tissue with the help of antibody dependent cell mediated cytotoxicity. So antibody dependent cytotoxicity, through this mechanism, these natural killer cells will damage or will kill these viruses. This yellow thing is there, no? That is the viruses and also tumor cells. This is all about the different cells which are mainly involved in the immune reactions. So here is a table which is showing differences between T and B lymphocytes. So the origin of both these lymphocytes is bone marrow, but T cells are produced from the thymus region. That is the reason they are called as T cells. As they are originated from thymus, they are called as T cells. When coming to B cells, they are originated from bursa region of bone marrow. So that is the reason they are called as B cells. Okay. In the lifespan, coming to lifespan, like small T cells, they'll be having a lifespan of months to years. When coming to B cells, it's less than 1 month. And T cell blast, from which the remaining T cells are produced for them, the lifespan is several days. Even for B cell blast, also the lifespan is several days. And then coming to location, T cells are mainly seen in a perifollicular region of lymph nodes and periarteriolar region of spleen and perifollicular region of Peyer's patches. And these B cells, they were seen in medullary cords, germinal centers, red pulp, central follicles of these lymph nodes, spleen and Peyer's patches. And in circulation, T cells are present in large amount, like they near about 75 to 80%, and B cells are present like in 10 to 15%. When again in T cells, these are of like T helper cells and T suppressor cells, right? So T helper cells are double the number of T suppressor cells. Okay, 2:1 ratio they are present, like TH and TS, they are present in the ratio of 2:1. And their complement receptor is absent, FC receptor is absent, antigen receptors, only antigen receptors are present in T cells. Okay, when coming to B cells, they'll be having the surface immunoglobulin because plasma cells will produce immunoglobulins, and FC receptor is also present, that is translating, which helps in binding of this antigen, that is present, and complement receptor, that is BCR, B cells complement receptor, that is also present. And coming to CD markers for TH cells 3, 7 and 2, T suppressor cells also 3, 7 and 2. When coming to B cells 19, 20, 21 and 23. And when coming to functions, as I said that B cells are involved in humoral mediated immunity, T cells are involved in cell mediated immune reactions. So this is all about the differences between T cells and B cells. So in this class we have discussed regarding T cells and B cells. I hope everybody is clear, and if you have any doubts you can feel free to ask me. Thank you all. Thank you everyone.